Okay, so last time we had talked about the non-covalent bonds and the way that different molecules interact with each other, and talked about a couple of important pieces there that the non-covalent bonds are pretty, even though they're weak, they are pretty stable. And so when you have a lot of them, it allows you to interact together in a stable manner. Another thing that also we'll see is that it's important these non-covalent bonds not be permanent. We want to be able to break these interactions and form new ones in real time as the needs of the cell and the things that we're trying to do take place. So all of those are important pieces to the nature of those non-covalent bonds. What I want to get into in this section is another concept that probably you have been exposed to before, and that is this idea of complementarity, or what we call complementary surfaces. Another property that non-covalent bonds have, or that give us inside the cell as these molecules begin to interact with each other, is a very large degree of specificity, meaning that when you think about these different bonds, so the hydrogen bond with its slight positives and negatives, and those positive and negative, full positive and negatives for ionic bonds, and the hydrophobic surfaces for hydrophobic interactions, all of these match up pretty specifically. And if we orient them correctly, then what we can do is basically manage who is going to interact with whom inside the cell in a very, very meaningful way. So in previous classes, you probably have referred to this kind of interaction as like a lock and key mechanism, meaning that one side of the interaction is the lock, the other side is the key. And what I want you to basically get to in this course is understand that what lock and key is talking about, be able to explain that at the molecular level, is these complementary surfaces within the context of those non-covalent bonds. So let's go back a little bit to last lecture's material. Throw up this picture here. So remember, this is the DNA double helix. We're talking about these complementary base pairs. So there's that word. And what you'll notice is that what complementary means is that these guys complement each other, meaning that they're not the same, they're a perfect match. They are the opposite to each other. And so here with these bases, G, A, T, and C, notice how along the center line here we've got all these O's and N's, these electronegative atoms. We've got these little hydrogens attached to one side, so that would be a covalent bond and it would be polar, and that would give it sort of that slightly positive. And then on the opposite side you have one without a hydrogen, and that would be a slightly negative side. So slight negatives and positives interact, and this is what makes T and A perfect match with each other, and G and C a perfect match with each other. Notice how if we tried to match up T with G, you'd end up with a little hydrogen there and a little hydrogen there. So they actually have the same potential for bonding, which means that they don't match up well, they don't complement each other well. So it kind of illustrates both sides of the relationship here. Now, what does this mean with regards to other molecules? Here's an enzyme called Rubisco, which is a plant enzyme, and basically what it does is it's looking for this protein called it's not a protein, it's a molecule called RUBP, and this is where plants get the CO2 from that they basically are going to use for making sugar during photosynthesis. And what we have here is this molecule coming into the active site of this enzyme, and of course the enzyme is made out of amino acids, and these amino acids have those little groups on them, the R groups that make them unique. We got several lysines, an arginine, a histidine, aspartic acid. And notice how these R groups are going to interact very specifically with groups, or even other groups like an ion, in the context of this RUBP. And so RUBP fits very snugly into this active site, and these R groups are going to match up perfectly with all of the different parts. So it recognizes RUBP and no other molecule. Our UPP is the only one that goes in there and complements it and fits, and then it catalyzes. We catalyze the reaction. So this guy will catalyze, and what we'll do is we'll break off that CO2. It gets put into that Calvin-Benson cycle, which builds it into the sugar. Okay? So when you think about this concept and the way all these molecules are going to end up interacting together, it really comes down to your potential. If you think about each molecule, even though they may be shaped exactly the same or built off the same building blocks or structure, what we actually attach to them based on its bonding potential is going to allow us to do several different things and recognize several different things. So things like receptors, recognizing their signals, enzymes recognizing their substrates, transporters recognizing their cargo. All of these molecular interactions can be very specific because these particular proteins are able to recognize specific molecules based on these non-covalent interactions. So here's one that's really quite appropriate for our course material here with regards to male and femaleness in mammals. So you guys are probably fairly familiar with these two hormones, estrogen and testosterone. Both of them are built off of the cholesterol backbone, so they're both steroids. You can see that shape-wise they're almost identical. So if we were trying to distinguish them just based on shape, we would not be able to. But you'll notice here, attached to this fourth ring, that we've got, in this case, an OH, and in this case, a single oxygen. And this is critically important to being able to tell the difference between these two hormones, because notice that these two have completely different bonding potentials with regards to hydrogen bonds. I'm going to see if I can draw on here. So if we have an OH, then obviously we're going to want some receptor that has the ability to come in and hydrogen bond with that, whereas in order to interact with this one, we're going to come in with the opposite. And so this ability to distinguish between this hydrogen bond and that hydrogen bond is perfect, it's specific, and it allows us to tell the difference between testosterone and estrogen, whether we're a receptor for testosterone, whether we're a receptor for estrogen. So those receptors may actually look very similar to each other, and the only difference is that at that location in this receptor, so we draw like a binding site here, then at that location, this particular one will recognize only estrogen, because if testosterone gets in there, what happens? Those are the same, right? It's kind of like G's and T's get together in the nucleotide, where the same charges, so slight negative, will repel each other, and testosterone will come back out. So it's a pretty important piece. Another place where we see this would be the recognizing even sugars. If you've had your organic or your the biochem even by now, you'll remember that the hexoses are going to have exactly the same chemical structure, and usually what separates them is the orientation of their carbonyl groups. And so here is galactose and glucose. They're exactly the same except for the fourth carbon here, where the OH is on the left side versus the right side relative to each other. Now chemically that may not make much of a difference, but in a cell, if I need to recognize glucose versus galactose, it actually is very useful, because you remember inside cells, sugars are going to create these ring structures. Okay, so where this O is going to circle around and bind to the C down here. And so what happens is that when we take the galactose side versus the glucose side and put it in a ring, for galactose, this OH group, which again is going to be able to hydrogen bond, and over here the OH group would hydrogen bond. Probably should have made that dotted line. And so in galactose, that OH goes up. In glucose, that OH goes down. Do you think an enzyme can tell the difference? Sure enough, just by putting its complementary piece, its lock and key, is going to recognize those OH groups very specifically. So these OHs, these positives and negatives for ionics, and these hydrophobic interactions, the ionic and the hydrophobic are not quite as specific as hydrogen bonds, but there is still that positive negative in those hydrophobic surfaces that come together in specific ways to allow certain molecules to recognize each other exclusively and not recognize anything else. And we do it and still be somewhat transient. We can release it or let go of it if conditions require us to do so. So it's a pretty neat system. Definitely spend some time trying to figure out what complementary means, be able to describe that lock and key mechanism that you've heard before in this way. Alright, so next time what we'll do is talk about some things that affect the way assembly takes place and go from there. See you then.